Our second speaker of today is Frederic Béen. And Frederic received his education in forensic science at the University of Lausanne. After obtaining his master's degree, he also obtained his PhD degree there in forensic science, and he focused on developing analytical methods and statistical tools to monitor drug use in wastewater samples at community level. And being very successful in doing so, Frederic continued testing the waters as a researcher in Lausanne as well, by setting up nationwide campaigns to monitor the use of illicit drugs, not only in Switzerland, but even up to Australia. And after working as a postdoc in Antwerp on environmental monitoring and wastewater, Frederic moved to Nieuwegein in 2018, where he, as a researcher, continued to work on sampling and analytical techniques to monitor emerging contaminants in the water cycle, mainly using chromatography and, of course, mass spectrometry. And at this moment, Frederic is managing international research projects to monitor all kinds of unwanted species, from small molecules up until viruses, I believe, in water for the benefit of human health. And today, Frederic will share with us some insights into how mass spec has allowed epidemiological monitoring of illicit drug use in wastewater. Frederic, I'm looking forward to it.
Thanks a lot. I hope you can hear me. I'm going to share my screen. Thank you very much for the great introduction. And yes, as you could hear, somehow I have developed a passion for wastewater, which sounds a bit gross, but it's actually a really interesting source of information about human health and lifestyle and behavior. And my talk today is actually, giving you an overview of what's been done in the field around analyzing wastewater, in particular for drug use and illicit drug use. So just briefly an overview of what I will talk about today. So give you just an overview of what we call WBE, so wastewater-based epidemiology, the whole discipline of analyzing wastewater. And then I will go through, really the beginnings, how it started off. And then, well, kind of perfect in line with the talk from Ruben, although this is more applied about the analysis of NPS, also in wastewater using screening methods and high resolution MS. And then I'll continue to something that is pretty relevant for the Netherlands, the fingerprinting of drug manufacturing through analysis of wastewater. And then, I will finish off with a few thoughts, personal thoughts about the future of WBE. So let me start with what WBE actually is. You might have heard of it. I'm just going to turn on my laser pointer. There you go. Oh, no, this was the wrong one here. You might have heard it. If you didn't hear about it before, you might have heard it since February or March of last year. It went from, I tend to say from niche to mainstream, in the sense that it has become a really big topic because it allows to monitor the circulation of the SARS-CoV-2, so the coronavirus, in communities by sampling their wastewater. But before before the pandemic and before the topic became so hot, there was already quite some research in the use of wastewater for tracking various kind of things. Among others, the most popular, I would say, is drug consumption. So the idea is that, well, as you know, in particular in developed countries, we're all connected to a sewer system, which conveys the wastewater, which is toilet flushes, showers, household appliances, but also industries and larger facilities are conveyed to a wastewater treatment plant, where the wastewater is treated before being released into surface water, so rivers and lakes. So what people do in wastewater-based epidemiology is that they take advantage of the infrastructure that is already there in terms of water quality monitoring. So we collect samples at the influent of a wastewater treatment plant, and then applying liquid chromatography most of the time, and obviously mass spec, to analyze these samples. We calculate, the concentrations of drugs or biomarkers in the samples that we collect. And we multiply these by the total flow of wastewater, which is something that is measured by the wastewater treatment plant themselves. And then we start working ourselves upwards. So using existing Well, using, first of all, data about how many people are connected to a wastewater treatment plant, we start calculating so-called population normalized loads. So it's basically the amount of metabolite that is excreted just per 1,000 people. And then including some pharmacokinetic data, we try also to do estimations of the amount of drugs that are being consumed by the people to give actually a significance in terms of really quantities. And eventually also you could do making a lot of assumptions in terms of the size of a dose and so on. You can do some estimates in terms of how many doses are being used. The interest of this is that, well, you can basically give a figure to the size of a drug market. And but what's really interesting and what's more accurate, I would say, because you rely less on assumption, is to follow trends over time based only on the mass load, so the amount of metabolite that you measure in wastewater. So let me walk you through all the things that happened in the last 12 years. So the first applications of this concept of using wastewater to track illicit drug use actually were applied not in wastewater, but in surface waters. So it was done by a group of researchers in Italy who monitored the presence of cocaine and benzoyl-agonine, its main metabolite, in the waters of the River Po. And basically the reason why they were detecting this drug is because they were it was being released through wastewater treatment plant effluents, which could not completely remove the drug from the water before it was released. So with this first attempt, applying some methods which I will walk you through in a second, they basically gave birth to this new discipline. Although conceptually it was already out there, in the sense that, well, there was a researcher from the US EPA, the Environmental Protection Agency, which thought about the idea of measuring biomarkers for population health monitoring already in 2000. But the group in Milan was the first one who applied it in 2005. And what they did, and this is still true actually nowadays, the collection of the wastewater samples takes place, as I mentioned before, at the influent or the wastewater treatment plant. For quantitative methods, there is always the addition, as much as possible, of mass labeled internal standards, not always available, especially if we're talking about compounds like the ones Ruben was just mentioning before, so NPS. And then there is most of the time a filtration step or a centrifugation step. And then two approaches are being used. SPE was very common, although slowly people are moving away because also of the increased sensitivity of triple quads. People are and also the high resolution MS instruments, people are moving more and more towards direct injection or dilute and shoot methods. But in general, mixed modes and cation exchange mixed mode phases or HLB reverse phases were often used for the sample prep. Then when it comes to the instrumental methods, well, most of the time, I would say most of the applications actually, especially initially, were looking into, well, we're using reverse phase chromatography as a standard method. But some groups actually also started including HELIC, and we do it ourselves, for example, at KWR for the analysis of certain types of markers of cocaine, in particular of crack cocaine. So once the product is being smoked, we have developed methods for the analysis of some of these pyrolysis byproducts in wastewater using HELIC. And there is also groups that have been recently reporting the use of mixed mode columns, also for the analysis of these smaller and rather polar compounds. Triple quads for the quantitative analysis, the most commonly used, although at KWR we actually do our quantitative analysis also with our Orbitrap. So we use for the screening and the quantitative analysis always the same instrument. The label standards, as I mentioned before, and the methods, yeah, already back in the first applications were in the low nanogram or sub nanogram per liter range, which, considering the complexity of wastewater as a matrix, is actually rather good. And yeah, as I said, back then there was use of SPE with 100 to 1,000 time concentration factors, although always with a delicate aspect of matrix effects which might come along. And then from the first applications with the conventional drugs, things actually really moved on, and people started realizing that basically, yeah, wastewater is a really gold mine of information in terms of substance use. So then from illicit drugs, people started looking also into consumption of, well, listed drugs, if I can call them like that. So alcohol consumption. And here you have an example of two studies, one in Spain and in Italy, and another one here where alcohol consumption measuring ethyl sulfate was measured across different cities in Europe. And what the researchers here also did was that they compared levels of alcohol consumption based on wastewater and compare these to alcohol consumption estimates from, for example, organizations like the WHO or things like that. And then, well, I had to squeeze in one of the things I had worked on when I was actually in Australia, also related to pyrolysis, the analysis of pyrolysis byproducts. So in Australia, there was and there still is a rather big issue with the consumption of crystal meth. And the group there has been analyzing methamphetamine in wastewater for quite a while. And when I was there with the researchers there, we thought, like, can we do something about and start looking into pyrolysis as well, because crystal meth is also being smoked. So we started looking into alternative or additional markers that we could measure, and we were actually able to detect dimethylamphetamine in the samples. Obviously, it's also an impurity of methamphetamine, so the interpretation is not that obvious, but we were able to see some differences in the concentration of the pyrolysis byproduct or synthesis impurity across different locations, in particular when it was with respect to rural and urban areas in different areas of Australia. And then moving on to the third part of my presentation, so the use of screening methods, and in particular for NPSs. I think I don't need to explain this a lot to you because you've already heard about it, but there are really a lot of NPSs out there. The European Centre for Drug and Drug Addiction, short EMCDDA, has a list, and they regularly add new compounds to the list. So these are all, yeah, so-called designer drugs, which, as Ruben was mentioning, are not submitted, or some of these analogs are not submitted to regulation, and people continue synthesizing new ones. So it's really complicated to trace them all, to analyze them, to know which ones are being consumed in the population. So this is where the application of, yeah, high resolution MS and wastewater analysis can play an important role. So as I was mentioning, there is an ever-growing number of these compounds. There is often a lack of pharmacokinetic data, so we don't really know what to analyze. And yeah, for the particular part of wastewater analysis, it can be tricky because the concentrations can be very low. A lot of these compounds are actually extremely potent and more potent than the original drug they're supposed to mimic the effects of. So there are a number of approaches and concepts which have been brought out there. And so this one is one that I took from a paper from 2014, where information about the early warning from the EMCDDA, to use that to try to using in silico and in vitro approaches to try to understand or identify which targets to look for, and then by using different approaches, which I will talk about in a second. So, for example, analyzing pissoirs at large public events when they were still allowed. This is unfortunately not happening that much lately. Or municipal wastewater treatment plants. And then to do chemical analysis and to, basically as a loop, try to continuously inform about the presence of these drugs in the community. So as I mentioned, wastewater analysis during major public events or the use of urinals. And here is where HRMS actually really comes into play as an additional tool to our analysis, because as I mentioned before, so targeted analysis, we can do them and they are extremely useful and sensitive. But on the other hand, we are limited in terms of the number of compounds that we can monitor, and we rely on standards. So suspect screening and non-target screening methods can be really useful in these approaches. And there are quite a number of suspect lists that are out there among forensic laboratories and researchers that share this information also through the EMCDDA, as I mentioned. So there are ways of creating suspect lists that can be used to actually prioritize and eventually identify compounds in the samples that we analyze. And this is really useful because if you're trying to understand whether a given NPS is being used in a community, of course, seizures can give you an idea. Testing streets, so people bringing in drugs for testing, also gives you an idea. But here you have an additional one because you're actually measuring what people consume. And then non-target screening also, of course, but you know, most of you are aware about the complexity and the time-consuming aspect that comes with fully non-target approaches. But here, just to give you some results of some research that has been done. This was carried out in the Netherlands. Was a PhD student that was working at KWR and at the University of Amsterdam, if I remember correctly, where with a suspect screening workflow, she was actually able to identify a certain number of compounds back then. So the analysis was done both with a QTOF and also with an Orbitrap. And she was able to indeed to tentatively identify certain compounds and even actually formally identify them with the analysis of reference standards that were available. And this was for wastewater. Yeah, these were actually wastewater samples that were collected during a major public event. Another approach is, as I mentioned before, the use of pooled urine samples. So also from urinals from large events. One drawback is most of the time these target only male population, which is probably the most prone to use of this type of substances, but this is one of the biases that comes with it. And the interesting part here is that, well, there is what they did in this study is that they didn't do any particular extraction besides protein precipitation, and then they analyzed the sample as it was with a QTOF. And I show you some example here of actually they were able in these samples also from a festival to identify alpha pyrovalerone and NDPV and two of their metabolites, as you can see here. So again, this is quite interesting, and these kind of urine samples are really ideal for doing these things because you're facing rather high concentration of samples compared to wastewater, which is often diluted, yeah, somewhere between a factor 100 and even more in case of rain events. So in terms of the uses and the applications, well, it's certainly an additional and complementary information about consumption at the population level. You get this temporal and spatial information, both for NPS and conventional drugs, which is harder to get if you're either doing surveys in the population, which have the drawback that people often don't know what they actually consumed when they're reporting. As I mentioned, there are some limitations with wastewater because it's highly diluted as a sample, but pooled urine and urine from festival or particular events can be an interesting alternative matrix. Of course, it's always biased because you're looking at it's a type of sample that you're collecting in very specific settings. So it's not necessarily, actually, it's most likely not representative of the general population, but it's still a population at risk. So that's quite interesting to monitor. And now the next step is related to the use of, in particular, again, high resolution MS for the fingerprinting of drug manufacturing. So being in the Netherlands, I guess you're all aware about the large manufacturing that takes place in the Netherlands. So here I just took, again, some estimates in terms of the amounts of drugs that are being produced, amphetamine and MDMA. And in both cases, there is a lot of waste that goes with it. So per kilogram of amphetamine, roughly 20 kilograms of waste are being produced. So if we take this as an estimate of the yearly production, we get to almost 6,150 tons. And then same things for MDMA, roughly 1,000 tons of waste. And the question is, yeah, where does all this waste go to? Obviously, they don't have a license, vergunning, I don't know what it's called in English, but a license to treat this waste. So it has to go somewhere. And I guess you're all more than familiar with this kind of pictures of, you know, dumping taking place either in nature or, yeah, this is a picture of an apartment where they tried to dump drugs down the toilet because the police was coming to arrest them. And another important aspect that puts everything in context is that since since the last 2 years, actually the number of locations where waste is being disposed have been decreasing. So obviously there is the question, where does this waste goes to? As I showed you before, 8,000 tons a year estimate. So it has to go somewhere. And one of the things that we're seeing in recent years, because people obviously are trying not to get caught, is the infiltration in soil or the disposal in in sewers. So one of the hypotheses that we had about the work that I'm going to present to you now is whether we can use the patterns, so the loads of MDMA and amphetamine that we measure in wastewater, to actually prioritize certain type of features which might help us detect disposal of drug manufacturing waste, and maybe even go one step further and tell us something about the synthesis route that is being used. So to do that, we use the pilot wastewater data from amphetamine and MDMA over the course of 3 years, as you can see here. So in red, you have the amounts of amphetamine and the amounts of MDMA that we measured in the wastewater across this period. And these peaks are actually what we suspect being dumps of synthesis waste, because if you back calculate the amount of drug that would correspond in terms of consumption, it's just impossible. Everyone from age 0 to 99 would be using drugs if that was the case. So it has to be dumps. So using a suspect screening workflow, using this data, so retrospective analysis that had been done on our Fusion Orbitrap and then our Fusion Orbitrap, and creating suspect lists of both MDMA and methamphetamine. So we included their products, precursors, pre-precursors, impurities, and so on. We wanted to see if we could find some information in these samples. So we started off initially with, I say we, but it's actually work that was done from a visiting PhD student that we had. So we started off with some conventional analysis. So of course, you're all familiar with these volcano plots where you try to see log changes in the intensity of features between what we call consumption and dump. So baseline consumption and dumping events. And we thought, okay, are there maybe some features in these ones which might be indicative of, you know, other information about the synthesis or so that was being used. And the same thing for these PCA plots. As you can see from the PCA plots, not particularly informative. So we were not really able to arbitrarily differentiate between the type of samples. So yeah, the conventional analysis was rather arbitrary. So how do we define a dump versus a baseline consumption in the wastewater? These are more details. So we didn't take into account flow, because the intensity of the features might change in the samples, not because of their concentration, but actually because of rain events or dilution of wastewater. So it's not really the best approach. So we tried to come up with an alternative strategy, and we had a premise to do that, is that we know that synthesis waste still has high, I put high in terms of with respect to conventional concentrations in wastewater. So there is still quite a lot of amphetamine and MDMA in the waste itself. So if you dump it, you're supposed to see it. So the approach involved normalizing the features that we detected, and then trying to measure the correlations between the intensity of features across all the samples that we analyzed and these amounts that we found in terms of MDMA and amphetamine. And we used here different types of distance measures to actually prioritize them. And this is what I'm going to show you here. So this is basically this algorithm that we came up with. So we try to correlate, for example, or measure the distance between the amount of one of the two drugs and the intensity, the normalized intensity of the features in a completely non-targeted way. And what Naomi, the students that we had, was able to do is that she started to find a certain number of compounds, both from the suspect list, but also fully non-targeted, that had quite high correlations with the amounts in wastewater. So she created a list that, yeah, some of these compounds for people that are commonly work with this kind of drugs are not so surprising. Safrole is used as a precursor, PMK as well. MDEA is an impurity, but some other of these chemicals, if we will be able to identify them, that's still work in progress, could be actually quite interesting to monitor, to give us an idea about the synthesis routes. So as I said, this can be used to track and detect illegal waste. It helps us to differentiate between consumption and waste disposal. And as I said, it can help to get an idea of what kind of synthesis route is being used, because obviously criminals are very inventive, very creative, so they keep on changing the routes that they use. And because some of the precursors, so the products that are being used to manufacture the drugs, are controlled, this approach can be used actually to add new chemicals to the list. But as I said, we still need to identify some of these. And this brings me to the end. I see I'm already a few minutes over. Sorry for that. In terms of the future, it's very short. So I gave you a glimpse of what can be done in wastewater-based epidemiology thanks to mass spectrometry. And I focused my talk about drugs, well, whether licit or illicit drugs, but there is really a lot more out there. As I mentioned, wastewater is really a goldmine of information about pharmaceuticals and personal care products, population markers, industrial chemicals, exposure to industrial chemicals, things related to stress, to food, to diet, habits. These are also things that can be mined from the analysis of wastewater. And then obviously, well, the last part, the biological, that's probably known to all of you with the analysis of pathogens and so on in wastewater. This brings me to the end. As I said, it's not only my work. There is a lot of people at KWR who are involved in the research that we did. Annemieke was one of them. She was also involved in all of this work. So I would like to thank my colleagues. And I would also like to thank the European Union for the support that they give us in the work that we do. This is a project that we have running on the analysis of drugs in wastewater. And thank you all for your attention, and I'll be happy to take questions if there are any.